I'd just like to welcome Mercy to our Sisonke Biotic seminars. We're really privileged to have Mercy here. I don't know if anyone's had a chance to take a look at her bio, but it is the length of my arm. They are a fantastic academic and a very prolific entrepreneur. Excited to hear what they have to share with us today. And also they've gone out of their way to meet up with us despite it being about 6 o'clock in the morning on their side of the world. So really big thank you, Mercy. And yeah, looking forward to the talk.
Yeah, thank you for having me, and thanks for everyone for showing up to wherever your time zones are. I realized the bio I sent out was a bit outdated. I'm currently a research scientist at Duke, but what I'll talk about here is mostly, I'm a research scientist at Google Research, sorry. But what I'll talk about here is mostly work I did during my postdoc at MIT and my PhD at Duke. And I'm really hoping to kind of demo some of the areas that we looked at in health and machine learning for global health, but also potentially provide inspiration and options for anyone who is still looking into ideas of areas to solve. So hopefully you find this somewhat useful. So just some background on me. From Ghana. I grew up in Ghana, and went to high school at Holy Child Senior High School in Cape Coast, and then in 2010 got a scholarship through the Zawadi Africa Education Fund to go to the University of Rochester in upstate New York in the US. So these are some of my friends from high school. That's my family, and that's Rochester. After majoring in biomedical engineering during Rochester, I went ahead to Duke, where I got my PhD in biomedical engineering and global health. Following that, got a postdoc fellowship to MIT, and after spent a couple of years, did there 2 years, and then afterwards found myself at Google as a research scientist. And this is in the Google Ghana research office, which is pretty cool. So my journey through AI kind of started from during my PhD research. I worked initially on biomedical device development, so I'll talk about that a bit more. But this images from this device led me into the world of medical image processing and using feature engineering and machine learning techniques, which eventually led me to looking at more deep learning techniques for medical imaging during my postdoc, natural language processing for clinical notes, and I'm currently working on responsible AI for global health equity. So have this intersection of medical devices, machine learning, and health equity. Okay, so just to get into it, while I've been kind of in this field, I've noticed that quite a bit of proposals for mitigating bias in AI start here with data, and biases in AI refers to when, of course, machine learning doesn't work as well in certain populations as they should, and provides unfair outputs, but they usually start from data collection and preparation, mitigations in the actual model development, mitigations in evaluating the model, and then mitigations in when you deploy it. And it goes along the lines of things like creating data sets with diverse patient cohorts, looking at error rates, techniques for debiasing, inspecting the design, and then monitoring post-authorization. However, I don't think a lot of these take into consideration things that may be specific to resource-constrained settings. For instance, limited access to medical devices that may result in missingness of data as well, simply because the devices aren't there, inaccessible and disconnected health records, resulting from paper storage methods that don't necessarily work how they do in the West, and disparity quality between medical devices that do exist, but also disparate use cases. So for instance, in the US, mammography is a screening tool. In a lot of countries in Africa, it's a diagnostic tool. And this has implications for transferring models, right? So I propose that mitigating biases should really start upstream from the medical devices themselves and the data generating devices. So improving access to devices for diverse patient cohorts, ensuring the features obtained are accurate for diverse patient cohorts. For example, optical measures that may not work as well on dark skin, and then being able to obtain data from these devices. So now I'm going to talk a bit about my work on how I've tried to do this on some scale and some other opportunities in my line of work to make devices accessible to healthcare. And part of this starts with the human-centered design approach, really understanding what the problems are with stakeholders before solutioning, so you have scalable impact. And so this is a bit, and part of this as developing accessible devices, and I'll talk about the color scope which I worked on during my PhD, then using those to develop algorithms, improving low-cost devices, and then future opportunities for health equity work. So yeah, I'll start on this. And so this work at Toolbox for accessible cervical cancer screening was done primarily during my PhD, and is currently being commercialized by a startup that came out of it called the Color Health Foundation. And I'll give some background, since I don't know if everyone is familiar with cervical cancer. It's a cancer that starts in the cervix, which is the lower part of the uterus, and it can take up to about 15 years to grow from infection with the HPV virus to full-blown metastatic cancer. So it takes a long time, and it's treatable if caught early. However, in a lot of low middle income countries, there's a high mortality rate. Greater than 90% of deaths from cervical cancer happen here. So there's 500,000 new cases each year, 55% mortality rate. It says 85% here, but it's actually increased. It's that occur in low middle income countries. And because women are simply not getting screened, there's no access to screening tools, and it's slow, preventable, and treatable. So why, and just to say, even in the US, in high income countries, there's still disparities. So like this is mortality for Black women in the US and mortality rates for white women, and it's more than double the rate. So why does this happen? It's because the management process is resource intensive, 3 different steps for screening, diagnosis, and treatment. And some of these are expensive. So colposcopy, which is used for imaging cervical cancer screening, is expensive and has, even though it's highly sensitive, whereas what the WHO usually recommends for Africa is visual inspection with acetic acid. And it's cheap and portable, but it's subjective, low accuracy. There's no data collection because it's pretty much looking at the cervix with your naked eye. So I developed the color scope, which allows for a lower cost per clone-free imaging, and then clinically validated that in clinical studies in the US and in Ghana and Peru, and then worked on algorithms for this to change the current approaches. So, yep. And then this is kind of the coloscope being used by a patient to view their cervix, and we found it to be comparable to the standard of care. It's had global reach with both the coloscope and another device that is used for cervical cancer screening, the pocket colposcope, that we also developed, and attained a lot of recognition. So following on this, we thought, what if we could not only reduce the costs, but also increase access to diagnostic interpretation? So now we can take images with our device. Can we reduce barriers to interpretation? So community health workers, for instance, could use with the algorithm as an assistive device. And the features that we're looking at for developing our models were from sources of contrast that applied to the cervix during screening. So acetic acid, which keeps normal. If the cervix is normal, there's no change. But if the cervix is abnormal, it turns white with some texturization. And then green light, which highlights blood vessels. So these are all normal cervixes, whereas this cervix here has a cervical cancer lesion. And then visual inspection with Lugol's iodine turns the cervix dark, but in abnormality, it turns yellow. And there were some challenges in developing algorithms for this. So we had the new device, there was a small data set. Most existing algorithms that had worked on cervical cancer screening required prior selection of a region of interest, used a single contrast, and were developed for higher end colposcopes. So the goal of this was really to develop explainable algorithms for end-to-end automated cervical cancer analysis, and combine different contrasts to improve diagnosis. So for this, the methods were really we collected images that had been taken with the coloscope and the pocket colposcope during screening, and then pre-processed them using specular reflection removal, denoising methods, and segmentation methods. Applied feature extraction methods to look at the texture and color, generated a subset of features, and input them into different classifiers, and looked at different methods to combine them. And the reason for this feature engineering heavy approach was really because of the limited amount of data that we had initially. Okay. So once we had developed this, we tested it using cross-fold validation, and found that with the acetic acid contrast alone, so these are receiver operating curves for the different contrast and the different combination of contrast. And then the black dots here are how clinicians performed relatively to this. So with the AUs, with the curve, there's the area under the curve, and the closer it is to 1, the better. So with acetic acid, the area under the curve was about 0.78. With Lugol's iodine, it was 0.79. And we're able to increase it by combining the contrast to about 0.85 plus. And also performed as well, and in combination better than the physicians who had also annotated the images. We're also able to look at how the classification scores changed from normal cancers to from normal cervixes to high-grade cancers, for instance, and looked at accuracy comparisons between the algorithm, which had an accuracy of about 80%, and then the clinicians who also tested this. Okay. So going on to the next thing, we continue to develop this out. We continue to collect additional data sets. So the original data set, results from the original data set I showed had 134 images. And in a year or so, we had almost 1,000 images, and 880 of them were useful from different countries. And so we started exploring deep learning models and for classification and segmentation. So we used YOLO to segment, version 3 to segment the cervix areas from the images, and then use the ResNet to classify them. And this, while the AUCs here show acetic acid, VIA again, the green lights, and then how the combinations work, then you can see here that we were able to also achieve really improved performance on combining the contrast for a holdout test set of 124 images. So these devices and getting feedback on the algorithms were conducted with collaborators in Peru. They were using it in, our collaborators were using it in a mobile van that they used to go out to marketplaces, communities, to reach women, educate them about cervical cancer screening and breast cancer screening. And we also tested this in Ghana. We're working with the Greater Accra Regional Hospital and Battle Clinic, which does a lot of cervical cancer outreach. So now I'll talk a little bit about Sonogan, which is work that I did during my postdoc, looking at how we could use AI to improve accessible medical devices. And the focus here was on diagnostic ultrasound. Diagnostic ultrasound has several applications. It's non-ionizing, and it's in addition to fetal imaging to look at babies. It's also used for tumor detection, for early screening of cancers, and it's also used for cardiovascular risk. And in the past 20 years or so, there's been huge strides in taking heavy, bulky ultrasound devices to very small, portable ultrasound devices that can just plug into your phone. And this, I think, has partially been thanks to the global health community trying to make these devices accessible. And they've really gained widespread use and have a lot of promise. However, some of the limitations here is that the high-end systems have much higher voltage outputs than the low-end systems. And just to show that this is, for example, an image taken from a low-voltage ultrasound device. And we are trying to look at features here at the 4 cm mark. And you don't see that as clearly because of noise, which is not visible in the high output image. So there are some considerations when using these devices. So the idea here was, they have power limits, and can we develop algorithms or use a software-based approach to translate low voltage looking images to high voltage images? And this would have applications in areas such as portable ultrasound, but also even beyond that for areas where there's FDA power limits, or there's a need for better imaging in higher penetration depths. And for this, we explore generative adversarial networks, which translate images between different domains, and collected data using phantoms, so using ultrasound phantoms and a research ultrasound device that allowed us to collect low voltage and high voltage images. And once these were collected, we did some data processing, looking at, so these were the original pairs, and then just worked on some ultrasound physics-based processing methods to get to an input that could actually be fed into the algorithm to demonstrate the differences, and that the algorithm could get signals from. And we used a classic pix-to-pix image model with a units generator and a classification CNN discriminator. And what we found was, so there are three images here. There's the image from the low voltage data. This is the high voltage image, and this is the model output. We found that the model was able to learn to really denoise the features of the deeper regions and improve on visibility for the low voltage image, almost on par with the high voltage. So we also looked at things like contrast to noise ratio and found them quantitatively, in addition to being qualitatively similar for the model output and the high voltage image. This is another image that shows the example, but also shows the limitation and not being able to generate tiny speckle features as well. So I've kind of discussed work that was done to develop accessible medical devices, to develop algorithms for accessible medical devices, and to improve and algorithms to improve these devices. And I just want to talk about what, from my perspective, some of the things I'm really looking at as opportunities for future work. And that is really in multimodal digital health. There's all these new tools that really have the potential to improve access to personalized digital health that could overcome a lot of the limitations of current clinical approaches. There's activity trackers, sleep trackers, diabetes monitors, reproductive health monitors, and portable devices, and even genomics. And there's also a lot of room for innovation in these spaces. So I think when we are thinking about AI health equity, we should really also look at how can we innovate or look for innovative tools that actually serve low and middle income countries, and find ways to get that data or put these devices out there, be able to get that data in a way that is culturally appropriate. And I have a mobile device here because it's so universal in our communities, and then build AI models on top of those for equitable health. So rather than starting from here, start from here. And some of the ways that I'm already trying to do that are through a startup that I founded called Gap Health, that's trying to use smartphones as a way to democratize patient data in West Africa. Kind of everybody in Ghana, it's very common for people to get paper printouts of your medical records. How could we empower patients? And then you have to store these records for a really long time. And the idea is, how can we empower patients using mobile devices to digitally store their records and even use with their permission and to improve their health? So that's something that I've been working with a team in Ghana on to, one, provide digital-based healthcare, but also enable digital patient-owned health data storage. The other area of opportunity is really understanding robustness in machine learning algorithms for this. It's for pre-screened populations in cervical cancer, but can really apply to any area, because when we take some of these foundational models and there isn't enough representative data in the African context, what are ways that we could look at fairness and machine learning biases, but also improve them from different populations? And then I am a huge advocate for reproductive health empowerment. So I just have to put this in there. And given that mortality rates for maternal health, cervical cancer, breast cancer are so dire in low and middle income countries, what can we learn about these disparities with new data collection tools? This is just showcasing a smart tampon that Next Gen Jane is a company in the US is working on, but how can we understand these disparities and also in combination with genetics and other modalities be able to improve reproductive health? And with all these going on and using the human-centered design approach to not just solution, but really understand the true problem areas and then build solutions that are scalable for those. So I've talked about creating accessible devices, algorithms that work with these accessible devices and future opportunities. I hope I welcome your questions, and I hope this was useful. And I'd love to thank everyone who's helped with this work. There are numerous people from my lab in Duke, the Center for Global Women's Health Technologies, the Jamil Clinic for AI in Healthcare at MIT, the Center for Ultrasound Research and Translation at Harvard, and Gap Health, which is my startup. So thank you.
Sure, thank you so much, Mercy. If I can ask our audience to please give Mercy a virtual round of applause. Really, just a superb presentation there. And I must say, I'm just so impressed by your end-to-end approach and your holistic approach in the space. I mean, so often one might be at a conference and hear about some work on AI in healthcare, machine learning in healthcare. And often when researchers get asked, so, you know, how are you going to implement this? Or, you know, what is this doing in like a real world situation? That often gets, you know, the buck kind of gets passed to, you know, that's someone else's problem to deal with. We're doing more like trying to beat some benchmark. And what I really like is just, you know, how, you know, boots on the ground, this approach has been. I'm really impressed. So in terms of questions from the audience, you know, anyone's welcome to put up their hand. You can turn on your video on if you like or not. Also, if you don't want to ask questions directly like that, you're more than welcome to ask in the chat, and I will read your question out for you. But to get the ball rolling, I will perhaps, I mean, I've got a bunch of questions that I'd love to ask, but I'll just ask one or two for now, and then we'll get some questions from the audience. So I'm really curious then, Mercy, and I'm not sure if I missed this, but are any of these solutions that you've talked about today been approved by the FDA so far? And if so, like what their process like entails for anyone who might, you know, see themselves journeying down that road?
Yeah, so the pocket colposcope, which is one of the low-cost cervical cancer screening devices we developed, was approved by the US FDA and has a CE mark. So that was a huge milestone for us. So the device itself was approved, and now we are in the process for the just because you asked was really there were some guidelines. I don't know the exact details because we actually had postback, so people who were in our lab after their undergrad who really looked into this and filed with the FDA. So it's not rocket science. We had done enough clinical studies to demonstrate it was safe, it was it worked well, and the materials that we were using was safe. So that's what we needed to show. We also were able to show a predicate, and that's there's already similar-ish devices that I use. So like there's a colposcope that's typically used for clinics. So that really helps with getting FDA approvals to show a predicate, but also to show that what you currently have is safe and works well through clinical, real-world clinical studies. And we are doing the same thing for the algorithms. So we've done a lot of the testing on retrospective data sets, so data that was collected during the initial clinical studies, and now we are moving towards prospective data sets and testing them on new patients. So that would hopefully allow us to generate enough evidence to get FDA approval and be able to implement that. But for now, it's a research. The AI part is a research tool. The other devices, such as the mobile-based electronic health records, has been screened by the Ghana Data Privacy and Protection Act. So we are trying really well to make sure we work within those rules and make sure we have the right approvals in Ghana. One thing I have to say, though, is just because something is FDA approved in the US and has a CE mark doesn't necessarily mean you can just translate it. So in Ghana, for instance, there's also an FDA where once you have these approvals, they also double check or and make approvals if you have all the documentation. But if you don't have a US approval or CE mark because you're in Ghana, for instance, you can just send them all your documentation and they will approve it. So different countries have different methods of approval.
Well, thank you. And that was a very comprehensive answer, and I really appreciate that. I really enjoyed the various pieces of information you threw in there. And while we're on the topic of safety, I'm going to, this is a question I quite like asking, but because you mentioned that explainability was an important aspect of one of your tools. And often when talking about like safety, the explainability versus interpretability might come up. And often I think, at least in the healthcare space, there are definitely some medications that we use that aren't entirely explainable, but we know that they are still safe to use because of clinical trials. So I'm curious, in like that kind of context, what your take is on, let's say, explainability versus interpretability for safety.
Yeah, and I think I can shed a bit more light on what I meant by that. So I agree with you. So when you go for an MRI or ultrasound, you don't need to know exactly how it works. You just know it works, and the doctors are confident that it works because of clinical studies. So I think that's really important for models to show real world efficacy rather than, it's easy to do holdout tests and cross-fold validation, but similarly to diagnostic devices, to do prospective trials to show that they work without, in the wild, that they work in the wild, that they work across different areas, and if they don't, to know which instances they don't necessarily work in. And that can be informed by those studies rather than simply by knowing exactly what the nuts and bolts of the model are. But one thing that is still useful that I think for clinicians to look at is potentially, so when I'll go back to this slide because this is where that's talked about, we looked at this to kind of give ourselves a check on how things are changing from normal to high grade and whether it makes sense, the way the model is working. So I think it's also good for a sanity check. It wasn't necessarily saying these are the specific nuts and bolts, but it's just does the classification scores make sense? The other thing that we looked at was transforming it to different, when we're doing the feature-based approach, different textures and colors, and again, a sanity check on whether the textures and color differences were actually highlighting what they were supposed to highlight, and if there was any signal that, you know, a black box model could potentially pick up. So it's easy to throw in data and just say, oh, I'm getting a 90% accuracy or 0.9 AUC or something, but I think it's important to at least have some kind of sanity check to make sure, explore the data a bit, explore a subset of the data and make sure it's in line with what you think it is supposed to do.
Oh, thank you. And with that, I'd like to try and encourage the audience. If anyone has any questions, just to raise your hand, and I might also just pick on some individuals there. I'm curious, Abraham, do you have any thoughts on the presentation?
Yeah, hello, Mercy. Hi, Chris. Thank you so much, Mercy, for the presentation. I think my network was a bit glitchy while you were presenting, and Chris, I already raised my hand to ask a question. So I didn't get that part well, but there was this part you mentioned when you started getting to like deep learning approaches that you used. YOLO V3 for segmentation, and then you used various for classification. So I was wondering, considering the fast advances in like YOLO, the YOLO algorithm, I think they're at 7 or 8 or 9 now. Do you think like there could be like let's say a high boost in the results that you got from the experiment with using like more advanced YOLO algorithms? And I've seen a lot of people choose units for like segmentation, especially when it comes to like X-ray images. Do you think that's also applicable to like your work? And like, how does that come in if you want to like use a different algorithm aside like the YOLO for this your work? So I just want to know like how robust you could use other algorithms. Thank you.
Yeah, totally. And I actually, so I'll answer that, but I also have a follow-up to Chris's last question to an example. So yeah, there's so many machine learning, the machine learning world moves so fast, the health world moves very slowly. And I think when we started working on this, it was before all the different iterations of YOLO had come out, right? And for us, an incremental improvement by maybe 0.2 or something was not necessary. We didn't think that would necessarily significantly improve the model, but it's definitely something that we can try out. And I think it was a matter of choosing, like testing it on units, testing it on YOLO. So there were a couple of segmentation methods that were tested and figuring out which worked better for this case. But I think it could probably be interchanged, but we did check units. ResNet, like classification models have also improved since this paper was sent to Photonics West in 2022, but the team had been working on it since like 2020, like in terms of collecting data, doing different pre-processing and post-processing approaches. And I'm sure there's probably like a 5% increase that the new ones could provide. But given that this was a kind of proven that the concepts work, and at that point was good enough, we published that in SPIE, but it's still ongoing. So I have graduated, the lead PhD student on just now, Erica, is still working on it. So there's definitely room for improvement. On Chris's, I hope that answers your question.
Yeah, it does. It does. I do have some other questions that I might
Okay.
like you to also see on. So maybe I didn't see that part of the presentation, but you made mention of the fact that okay, this is now like a startup. Like you read the startup around this after your PhD.
Yeah.
So I was wondering if like what was the clinical like testing phase of this your whole experimentation like before you were able to like okay, yeah, I think it is good enough for me to say it's a startup. What benchmark did you use to say okay, my accuracy like this is fine enough for me to like push this for a startup, and the clinical testing trial for you, how was it like?
Yeah, so the startup has two areas. So it's the main focus is the device, actually making sure cervical cancer screening is accessible, and that's been FDA approved, widely tested, and that's really what it's running on. And we've done this work that's shown that retrospective data, so that's previous data that we collected during the clinical studies, can give us AUCs of 0.87. And now we are doing, we've just got funding to do prospective studies in AI. So the benchmarks usually would be that we, and since I'm at Google, I'm not as involved in the startup anymore, but the benchmarks would be to collect the data, have it provide us, rate what their impression of the cervical image is in real time, or of the cervixes in real time, have a cohort of providers, have a group or have a group input or majority vote on what the impression is, and compare it to how we perform. And what we want to do is not say we are better than, let's say, the gold standard, which is pathology, or as good as, but that we are at least as good as the providers who are expert providers with over 10 to 15 years of experience. So there's confidence in potentially task shifting some of this work from providers who might not be as readily available to community health workers who typically carry out the screening. So I wonder if that answers. And so that gives us evidence to collect FDA approvals for the algorithms themselves. So in short, no, this is not currently used. It's used as a research to the algorithms, and we are still collecting data.
Okay, no problem. Thank you so much. I really appreciate the answers.
You're welcome. Yeah, and then Chris, sorry, I meant to give an example to your question on interpretability and explainability. So part of the ultrasound work, actually, we when we started out, we had a data set that had been fabricated using well-known ultrasound methods and for computational methods for low voltage, high voltage. And when we ran our algorithms, we were getting 0.9 plus AUCs. And my colleague, who was very much an algorithms person, was like, this is great. We don't have to do anything else. And we are set. And we went a bit deeper, because as soon as that happens, that's, in my opinion, you just have to look deeper. And talking with ultrasound physicists, we realized that it was just a very easy problem using these computationally generated data for an algorithm to solve, but it would not be as easy a problem for real world data. So then had to scrap this and then go and collect new real world, albeit phantom data. And that was a lot more of a challenging task. So again, it's important, even though you might not know exactly what the model is doing, to at least sit down with experts, figure out what it might be doing, and just, again, provide a sanity check that goes beyond just the AUCs and the accuracy.
Sure, thank you. I really appreciate your answer there. And I'm also just aware that we only have about 5 minutes left, and we have some questions in the chat. So I think the ones that have already been asked in the chat are probably all the time that we're going to get for. I will invite those who have asked in the chat to unmute your mic if you'd like to ask your question. So we'll just go in order. Deborah, if you'd like to ask your question, and then thereafter Anandyat. And then I think we'll be, that's all we'll have for today. Unfortunately, Dina, I don't think we'll be able to get to your question, but let's see what we have time for.
Okay, so I think the first question on interpretability and explainability, I think you sort of touched on it a little with regards to the question I think Abraham asked. So I just wanted to know if your team has sort of explored or considered already trying to like really explain or interpret these models that you have. But I think you already mentioned that yes, it's important sitting with a team of experts and all of that. And the second question is with regards to fairness. I think at the beginning of your presentation, you sort of compared how the identification of cervical cancer is with Black Americans or something. I don't know if I don't know whether it was slide 12 or so. I'm not very sure on the number, but you had this plot, yes. So my question is, why do we have these disparities?
Yep. Okay. So the disparities are similar to why we have the kind of similar and different to why we have them in Africa. So cervical cancer, it's really a systems issue, and that's why we are really trying to solution this from a systems approach rather than just an algorithmic approach. So in the US, so cervical cancer is really easy to use. You go for screening every 2 to 3 years. You do you get your vaccine, your HPV vaccine. If you don't have, for everyone on this call, if you don't have your HPV vaccine and you can access it, please get it. But it helps prevent it. So that's the first step to blocking to preventing cervical cancer. Then every 2 to 3 years, there's screening that is recommended, just preventative screening. You don't have to show symptoms. You're going for screening. They check whether there's any potential malignant cells, and if it comes out as positive, then you go for diagnosis, and they confirm, and then you go for treatment. And it's really resource intensive. So one, if there's like it's socioeconomic factors, right? So if there's less access to healthcare, and healthcare inequalities, then it exacerbates cervical cancer. Because what happens is, and healthcare awareness too, because if you don't go in for this screening, it's asymptomatic. So by the time you start showing symptoms, let's say bleeding, pain, it's really quite advanced. And that's what happens in a lot of countries in Africa, where we see a lot of advanced stages that are really difficult to treat and also have a much higher mortality rate. And to a certain extent, that's also what's happening in the US. Does that answer your question? So,
but these mortality rates among these races are still different, and so like this brings me to the issue of ML fairness. So is this something that has to do with the biological makeups of something that is causing these differences? Like, have you like looked into why, aside the fact that probably people don't go for checkup often, but aside that, are there other factors that is causing this huge disparity? Because I consider that these people are all in the same country and have access to the same medical care.
No. So that's not the right consideration. So I think maybe, and that's my fault for not being clear on that. No, so in the US, it's very, very, health disparities by race due to socioeconomic status is something that's a huge public health concern. So same country, not equal access to same healthcare. So I hope that, and so it's mainly a socioeconomic status disease. It's not really there's not been any proof that it's genetic. There's genetic factors that cause these fairness issues. And in fact, a lot of high-income countries also had high cervical cancer incidences, and it's drastically reduced thanks to the vaccine, thanks to screening approaches. But within these countries, there's still disparities because not everyone has access. So there's definitely an access issue. Yeah. Does that make sense? So does that explain it better?
Yeah, that makes sense. Yeah, thanks.
Sure. So I'm just noting that it is at the top of the hour. So Mercy, if you do have to drop off, thank you so much. Just checking in. Is there time for one more question, or or is it definitely a hard cutoff? Yes.
Oh yeah, I have time for one more question. Yeah. Sorry.
Perfect. Okay, sure. Then yeah, this will be the last question, and then we'll bring the meeting to a close. So yeah, so you're Anajiyat, if you'd like to ask your question.
Oh yes. Thanks. Sorry.
Curious
Yeah.
that what was the size of the data set and the split of test train validation, and if K-fold or other techniques were used. Thank you.
I'm sorry, I didn't quite catch your question. There were two people talking actually.
Sorry. So I wanted to ask, what was the size of the data set on which it was trained, and the split between train test validation, and if there was K-fold or other techniques were used?
Yes, yep. Okay, so on the very first feature engineering approach that I showed, this was on a very limited data set. So that's kind of the challenges in developing algorithms for new colposcope. We're actually collecting new data, and I'll kind of talk about future progress since that. So here we had 134 images, which is super low. And after a year in different clinical studies, we had almost 1,000 images. And that was what was, so because we had 134 images here, we didn't use a deep learning approach. We used feature extraction and logistic regression, support vector machines. The problem with those is, feature extraction is, I don't know if anyone here, is feature extraction intensive or not, but you actually hand engineer what the physicians are looking at. So they look at changes in color, they look at texture. So you try to extract texture manually from the features, which is very intensive, and you try to extract color from the features, and then you put them into a logistic classifier. So it's nice because when you have limited data sets, it's a nice approach, but it can be very intensive to do for larger data sets. So once we had closer to 1,000, we had about 880 viable images. We started looking more at deep learning models. And here, so for the feature set, we used cross-validation to test again, because it was 134. For the deep learning models, we did I believe it was a 70-30 split. So 70% in training, 30% in testing of the 880 images. And then it might have been 80-20 actually. And then the test set was 124. So this was a holdout test set that the model hadn't seen. And it was actually a test set from a completely different set of clinics. So we trained with images from one set of clinics, and then we tested on new images from a different set of clinics. Does that answer your question?
Thank you very much.
You're welcome. And now what we are trying to do is we are getting more data. We are trying to do prospective studies where we are actually collecting new data and looking at how the algorithm works in real time, and trying to improve the algorithm. So I know there's data sets that exist. There's larger amounts of data sets for different devices online. We are also trying to see if those could be used as a to facilitate training.
Yeah, thank you very much. And thank you for going a little bit over time to answer the last of our questions. There's all the time that we have for today. If we could give Mercy another virtual round of applause. Thank you again so much for making the time to chat with us. It really was just fantastic. And yeah, I think, if anyone hangs around Sisonke enough, you'll see that Mercy has been contributing to some of our projects. I encourage anyone who's interested in any kind of collaborative paper writing to jump on to our collaborative survey paper project that we're writing, as well as keeping an eye out for other papers that we'll be writing together in the future. But yep, thanks Mercy, and thanks for everyone for attending. We will be posting this video up on our YouTube channel, hopefully sometime early next week. Until then, it's great hanging out with you guys.
Yeah, thanks for all your wonderful questions. Very interested, and it was great being here. So thanks for having me, Chris, and the Sisonke community.